lives in a skeleton, and it burrows and undermines the skeleton and makes it very brittle. And in some cases, the species of worm is diagnostic to the species of coral. But during that undermining process, it does make it very brittle such that they can fall under their own weight sometimes. But I'm not seeing a huge accumulation of debris here. So I'm wondering if this is just a very stable environment for them to persist in.
As opposed to them not having that worm?
Well, I think that the worm is consistent. I mean, even worldwide across their distribution, Hemicorallium, they all have polychaete worms associated with them. The question is, how much of the colony is undermined or weakened.
So here's a question for the entire group. By the way, how did you end up working among marine biology / geology and research related to it? Did you always know what you wanted to do or something like that? Or was it more of like a happy accident?
Mine was a happy accident. I was in geology at UNLV in Las Vegas, and then I started working in a geochronology lab, and just so happened Kevin Conrad, who's also on this cruise, was working with marine geochronology, and yeah, sort of connected.
Yeah, my background is, oh, actually, can we take a tight zoom on this colony right at the branching point?
Sure.
Yeah, so my background is largely biology focused. I did my undergrad with marine sciences, and then kind of specified into marine biology and benthic ecology. Yeah.
What's that? Why does it have an extra branch like that?
That's a good question. I know someone who is investigating that exact question, so that's why we're going to take a tight zoom here to figure out what's going on. So this looks like it's branching right above the node. Can we make a C-log entry with the word sparse brancher in here so we can find this observation later?
You can try and
Cool.
tighter zoom there.
Yep. Yeah, branching right above the node, or possibly at the node. All right.
Good.
This is good. Yep.
Okay, go away.
Steve, here's a question for you regarding your background. Steve, have you wrote any papers where the average Joe can find them? That's not a scientist / a professor?
Yep. All of my
squid.
publications, all of my publications are open access. So that means anyone can find them on the internet, free to download. Actually, one of them might be, well, there are versions that you can find, even not at the journal's website, that are free to get. We try to publish in open access as much as possible.
10 meters.
Although it gets very
5
pricey.
meters at least.
Sometimes the publication fees for open access are in excess of $4,000 or $5,000 per article.
Wow.
But it's important that that information is out there and available. And wherever I can, I try to make them available as PDFs. Huge Polyapgon sponges.
Challenging one, eh, Dad?
A little bit.
Big white balance in the middle of the
Yeah.
picture.
Just trying to see, there's so much stuff in the background too.
Okay, I'm gonna try and come down the hill a little bit more towards.
So what's going on with this bathymetry? Maybe folks in the front row can help me understand. Is there a ridge that goes up to the west, or is it just kind of go north all the time?
Seems like it's going north. It's running north, and then to the west there's a bit of a downslope. Is that what you're seeing, Dan?
Totally.
Yeah. Which
Interesting.
we don't see on
It's
our
tragic we're
maps.
not coming up the slope.
Well, there's got to be a top somewhere, so we might as well find it. We're this far already.
I can't get around.
You want to keep following the
yeah,
ridge?
we're totally going the wrong way. To go. If you want to go to the top,
I mean, the top of this local area to the west, right? Presumably it falls off again, right?
I'm looking northeast now, so west is downhill. Uphill is east.
All right. Well, cool. Can we drop a target here and say, you know, coral garden extends north and northeast, and then let's turn to the west and start heading up the ridge adjacent to waypoint 5.
That's what it looks like to the west.
Yeah. I think we'll just have to move a little bit, maybe 100 meters or so probably before we start to see any slope.
Yeah. Let me. Sorry,
I don't want to put us too far behind because I know that we're trying to get some rocks from further up the slope, and then we're trying to reach the carbonate cap eventually sometime during this dive.
Okay. Do we want to head more west then? Toward our original waypoints?
Yeah, I wouldn't go straight to 5. I would just go
Yeah,
straight up the axis of the ridge, yeah.
Yep. Okay.
Yep, okay.
Got it.
Can you?
Then
Can
to
you
answer
come
that
down
question
for a minute?
in the chat, the sponge on the right of the ridge, is that two
get
separate
turned
ones with
around.
coral in the middle? Yes. Yes, it is. Or it was.
Nice little knoll, yeah. Really nice
Come on,
spot.
come around. There we go. Sorry, I got tail to tail there. I couldn't come around.
Oh wow, this watch has flown by for me. This is I think that's a Faeria sponge. I
Yeah.
haven't seen that one yet.
It's a couple.
And answer the question in the chat, we're English students from Uruguay. Oh, South America.
A pancake.
And we are
urchin.
working on underwater mysteries. Where'd you go? What do you do on the Nautilus? Well, I am a student intern, so I do a little bit of everything. So I'm currently right now the Science and Communication Fellow for the 4 to 8 watch, as well as I help work in the wet lab whenever I can, and also help work with ROV launch and recovery whenever I can. So just a little bit of everything.
I'm Brandon. I do science, focusing on geology. With that, I cut some rocks earlier, or I guess yesterday. Talk about rocks,
Current's pushing me. That's
think
what's
about
happening.
rocks,
You
dream
are
about
a rock.
rocks.
Even this close, I can't get around with that
Just-
thruster. It's just
Yeah, the current is, now the current's to the north. Okay, I should be right under you now. You
Yeah, I
can
see
take
your
that
lights.
turnout.
the 6-8 turnout, or
Take your turnout, yeah. We're getting up there to the turn
Wow, look
limit.
at that
Beautiful
crazy walteria. Yeah, those projections coming out the sides are called lateralia, and oftentimes we find them covered with brittle stars or sometimes crabs like you see there. And then occasionally you'll see on those spines predatory sea stars too.
There's a good amount of color variation in the squat lobsters. They seem like white, yellow, orange.
Yeah, there's a lot of variation. We try to collect
Which
them
way are you gonna go
you know,
now?
with animals like corals as
Roger.
possible, but they're very difficult to sample otherwise if you don't take the coral
Bring
that
your
they're on.
head around the 260.
you want me to go around once more to get the other wrap out first, or just sit tight here? 260.
No, you'll have a half wrap at 260. That'll be good.
And then for
Gotcha.
Brandon, speaking of rocks, how has rock chopping been going? Have you found anything interesting, unexpected, or have you found anything that we are specifically looking for?
Going good.
Going well.
Yeah. Yeah, we found some rocks that we've been looking for. So we're looking for basalt, unaltered, if possible. And if not, then if there's good mineralization, even if it's altered, it's still usable.
Heading 260.
We found
Roger.
some good basalt and also some altered stuff, but it had good chronoproxene and amphibole in it, so still usable. So pretty good. As far as unusual, no, but I mean everything looks interesting. Yeah, we found, you know, a breccia, found pelagonite, found phosphorite, which, you know, they look cool, but we're not going to utilize them in our research.
The sun star there.
You can come down
So
a
last
few meters.
arid.
There's enough current to blow the tether out behind us.
Down.
Zoom in on the star if you want.
Sol asteroid.
Sol asteroid, yeah, like sun.
Yeah,
Sol.
clever.
Go a little tighter.
Yep.
Sorry, Steve, what's this called again?
This is a Sol asteroid, a sun star, a 6-armed star. Or 7-armed.
Yeah.
Huh.
One's dwarf.
The ones
Armed.
we usually see were, yeah, 6-armed.
Do they have 2 for backup?
I think most Sol asteroids
Okay.
have at least 5 arms, but I think they're usually more.
Ooh.
Here's another question from chat. Have you seen any benthic centrophores or on the walterias like that, or seen them in general?
Yeah, we see them generally. I haven't seen them on this cruise. But yeah, you'll find benthic ctenophores, a genus called Teolfiella.
Then answer the question in chat. Can you personally keep some of the things you find? No, we cannot, unfortunately, especially around here. This is a wildlife sanctuary, so it is illegal in the U.S. to take from this area, as it is protected by the government. So, I mean, you can, it's just very, very much not recommended.
The proper way to do it would be to get permits for any activities within the monument.
Is it hard to get a permit?
Well, you have to write to the permitting office and explain what you're going to be doing. And then they will explain your, for example, if you're doing science, explain your scientific needs and goals, what your objectives are, how you are, minimizing your impact, and they will grant you a permit for certain activities if it is deemed appropriate. It takes about, all in all, it takes about a year, maybe longer to go through the permitting process, so it's not a trivial thing.
Zoom in there if you want. Yeah.
I see.
Yeah, what is that?
Checks are good, comps are good.
Is that another
This
soul
almost looks like a
Heading 220 with
pythonaster or
Atlanta
something.
right now.
So this is a different type of sea star. It's not a, I don't think it's a sun star. I think it's a pythonaster sea star. These are actually known predators of, can we get a better close-up of this star?
Sure.
These are known predators of sponges, actually.
Oh.
Yeah.
Is it on its way to eat that sponge right next to it?
I don't know.
Scheming.
Run,
There's
sponge,
also,
run.
yeah, another smaller brittle star. These kind of pentagonal brittle stars,
Oh, yeah.
just to the top there. They're pretty neat animals too.
Good.
I think we're all set. There are very few sea stars that I feel confident in identifying, and I usually can get pythonaster.
Big piece of tumbleweed.
You think so?
You're asking me?
Oh no, I'm talking.
Okay, sorry. Thought you were mocking my
No.
tumbleweed.
Don't worry, Brandon, I'll mock you. Can you, another question from that, from chat, can you keep things from non-protected areas? Not entirely sure, I think so, but I don't think it's recommended by the
I
crews
recommend
NLS.
you don't.
Yeah,
Just
Brandon
leave stuff how it
recommends
is. Just take your picture and move on.
So Steve, someone has a question from chat. What was the green thing above that sea star? That's triangular and green?
It reminded me of the stuff you called sea mucus last time, Steve.
Yeah, I didn't catch that green bit. Yeah, there is a lot of mucusy producing stuff down here.
Another sea star, small one.
Where? Oh, is it in that middle?
Yeah.
Oh, okay.
There he is, yep.
Oh, this looks a little uplifted, kind of. It's weird.
Yeah. It's been a while. If you want to look for a rock, I don't know if there's anything useful up here.
Yeah, I'll look. We've been kind of up in the water column, so.
Yeah.
Another question for Steve. How do sponges reproduce and spread on the ocean floor?
Sponges have both asexual and sexual reproduction, so they can split off chunks and colonize new areas. You know, for example, the fragment that we saw, the tumbleweed, if it gets stuck in a spot, it can reattach and regrow. Otherwise, they can propagate. You know, larvae can land on a distant area and colonize that way too. I think they're largely planktonic larvae.
So when you say that they can sexually reproduce, does that mean there are male and female sponges, or are they hermaphroditic? Or hermaphroditic? Hermaphroditic? I don't know, something like that.
You know, I don't know if they display both sexes or not, or what if it's sequential or what the rule is for sponges. To look that up.
Homework for people at home.
That's why we need the sponge scientists to come in.
That
Talk
green
about
one on
you.
the right looks like a little Christmas tree.
The thing is about a lot of biology is that they're very adaptive, and so what happens is you know, they might have multiple different types of you know, reproductive modes sometimes within a single phylogenetic group, like a single phylum or a single class. So it's not clear.
You bring your head to the right a little bit.
You know, if one
Roger.
if one blanket statement would fit all sponges, it's probably a variation of things.
230.
Ah.
Big slab. Sea cucumber.
Ooh.
Well, there it is.
Still very curious about those crusty flakes we saw, those barnacle plates.
Crusty flakes.
But we haven't seen any of the barnacles yet that typically produce them. Usually we would see live barnacles too. And the thing about barnacles, they often grow in fairly high densities along, you know, the edges of rocks in these types of habitats. I haven't seen a single one yet.
You think they're way up there on the ridge, though?
I don't know. I think if they were further upslope, we would see them in many of these pockets, right? But they were really localized.
I see.
Now we've got these kind of like pseudo-nodules again, or basalt fragments.
The ones we scooped yesterday, were they actually nodules?
Have not cut them open yet.
Ah, okay.
just going through and feeling them, some of them feel heavy, but some of them are pure crust, very light chunks of irregular shape, just big chunks of crust.
I think we also scooped up a shark tooth in that pile.
Really?
Yeah.
I haven't seen it yet. I really want to go see it.
Yeah, it should be in the bucket where all the nodules are.
Nice.
We should isolate that so it doesn't turn into shark tooth powder.
Here's a question. Cal asks, when witnessing an unfamiliar, possibly new species in the sea, how do you go about identifying it? Do you have a field guide, or do you typically have to consult a database after reviewing footage?
Bridge nav.
It's
Can we move 20 meters 240, please?
It's a little bit of both.
Thank you.
So there's been a fair amount of exploration that's been done out here over the past 10, 15 years by outfits out of Hawaii. Hawaii Undersea Research Lab for a while conducted a lot of research and exploration using submersibles out here around Johnston Atoll, but never really at these kind of guyots and peaked seamounts. But more recently, since 2017, there have been concerted efforts by NOAA as well as OET and other entities to create or provide resources for photo field guides.
Maybe grab a rock.
We have one.
If we're not too far behind.
You want a rock, Brandon?
Yeah, this guy. Or maybe this.
Yeah. Sure. Go ahead, get that thing out.
Oh, pancake.
Zoom in there.
So bender and ground fault sense off. And then craft
What
power
is that
on.
guy?
Waiting
Is
for
that the
comms.
pancake guy?
Yeah.
Cool.
Yeah, it's one of the ones that Liz collected earlier this cruise.
I want to see him spit out sediment again. Or I guess poop out sediment?
Yeah,
For
fecal
a minute
material.
that you said cinnamon. I'm
What
like,
is,
oh.
Cinnamon.
All right,
Hold on
I've
a
got valid
second.
comms.
What is this?
Okay.
Can you
Wait
circle
a
the
minute.
rock
What
for
is
me
that
again,
thing?
Brandon?
Can we
Oh,
look
yeah.
at that too?
You want to see
Look at
that?
that.
Okay, take a look at that first.
This thing. What is that?
Is that
Zoom
a?
right in there.
That's not a.
Or is that full zoom
native
already?
Yeah,
Break,
it's
is
full
it?
zoom. It's not a nudibranch, is it?
It's teeny, whatever it
It's
is.
tiny, yeah.
It looks like it has a rhinophore of some sort.
I can't tell if it's moving on its own or if it's moving because of the current.
Current?
Yeah, kind of looks like a current. All right, let's go for the rock then.
All right, give me,
Maybe we
give
can
me
look at
that
that
rock
later.
circle.
What's that?
Which rock?
Which rock? Where were I? I don't remember where I was.
We can try some.
Okay, let's just try this guy.
All right.
Came off swift.
It must be in a continuous wrist there. Or I mean, position wrist.
Yeah, okay, I turned on continuous. All right. Right here?
Yeah. If you could show me. I'm not sure what it looks like yet.
itty bitty in there.
No, that's not.
Nope.
No.
All right.
Let's see if there's. You see anything angular, Steve?
I can move a little bit.
I can't tell if these are just like collapsed crust bits, but maybe these up here, or we might have better luck if we can move upslope a little bit.
I'm not seeing anything else in this area.
Too strong. We might be stuck with this rock, guys.
That's funny.
I need a giant toothpick.
I'm telling you, that battle saw. Left-hand battle saw, right-hand claw.
Then really going to answer that question. Are there types of coral, sponges, etc.
that
you can find anywhere else in the world, or are there any that have been found at a single spot and nowhere else? Well, Zed, it depends from place to place.
No, that's 22, but
sometimes you can find very, very common.
Which rock do you want, Brandon?
I'm looking. I'm not seeing anything.
Okay, that's it. We're getting drugged.
Yeah, we got to come up.
So it really depends from site to site. On one site, you could see a sponge that's super, super common.
Oh, you got a rock.
And then from another site, you could see a sponge that's super, super rare, but it's very, very common at a different site. So it really depends from site to site. And I don't think we've found any endemic species of sponges and corals for one specific area. So not entirely sure. Has the team ever seen a large fish on cam that either surprised or scared them? I mean, the Chaunacops was kind of surprising to see. Just seeing it floating there, kind of frowning at us. But I don't believe anything has quite scared the team. Am I right in saying that?
Finally found
No?
the uphill.
We're a pretty fearless bunch here on the EV Nautilus. So is that urchin more flexible or soft than regular urchins? It looks very floppy when they collected it on the other two dives. We're not entirely sure since we didn't really collect it, but generally speaking, usually yes.
Yep.
I'd say these urchins are pretty floppy and non-structured.
They're very wiggly.
Very, very wiggly.
Should be good for a minute.
Glad we got that eDNA sample when we did, because it's turning sparse again.
Here's another question. What sort of flora and or fauna would you be most surprised / excited to see on this expedition?
I need a break.
Most surprised.
Well, the only flora down here you're going to see is microbial, so I guess that would be interesting.
Ooh.
You wouldn't see plants necessarily. Sometimes we see like big, rafting plants if it's shallow enough, accumulate on the seafloor, but I've never seen it on a seamount like this. In the Atlantic, you see it all the time, like sargassum on the seafloor, but it's dead usually by the time it gets down there. Big food source though for sea urchins and cucumbers, things like that.
Spin around some way.
Then to answer the question, hello from Santa Barbara. Hi. What features are you looking for in a good rock sample? Well, from what Brandon has told us so far, specifically angular rocks
You ready to
with
move?
sharper edges and not too thick and easy to cut through.
Okay.
With good-looking ferromanganese.
Chaunacops.
Chaunacops Part 2!
There we go.
Yay!
Uh-oh, he's not happy about that.
I think we're a bit loud.
Yeah, he heard us yelling.
The way he swims is so adorable.
Or maybe he's coming to say hi to us.
Part of
Look
your
at that.
Yeah, I don't know. That frown says otherwise.
Hey, buddy.
Oh, okay, bye.
Oh. So this is what we were talking about earlier when we said the Chaunacops, or the coffin fish as it's also referred to. And this one seems to be a little bit more orange than the red one that we found. But
yeah. This is a little more typical color morph. Yeah.
He's an ugly little guy, isn't he?
I think he's cute.
Well, you know, sure, to each their own, I guess.
Face only a mother could love.
Yeah.
Any parasites on this one? Looks like it's got some sediment.
Are
stuck
those
to the bottom?
Are those fins or little feet that it has tucked away in the bottom?
Yeah, they're modified fins that act like feet with air quotes. Yeah.
Wow,
They're really just fins.
That is so cool.
Hello,
good look at its underside.
Wow.
Yeah.
Gorgeous orange eyes.
Wow.
I've never seen one swim this far up before.
Ah. What are those fleshy protrusions that he has like along his body?
Fins, or you mean around the mouth?
Or like everywhere, like it kind of looks like, you know those spikes on a pufferfish or something like that?
Yeah,
Yeah.
I think they're actually sensory projections.
Ooh.
So it uses them to detect vibrations maybe nearby. Because they're ambush predators, they have a big mouth.
It's
They'll
full wide.
sit and wait until prey comes along, and they'll open their mouth and they effectively create a vacuum that sucks in
It's like a slurp.
their prey. It's like a slurp, yeah.
Nice.
Do you want the lasers on top?
Is it kind of like a wabagong, Steve?
I think so. I don't know much about their biology, but yeah, I think they sit on the bottom, right, and they camouflage. Yeah.
Wow.
These probably are more active. You will use their photosynthetic lure to maybe either photosynthetic or chemosynthetic lure to attract prey items. Oh, no, not photosynthetic. What am I saying? It's time for coffee again. Bioluminescent, sorry.
And then here's a question for Steve. Do you share data you collected with other researchers in other fields like space, volcanoes, or above the sea?
Well, all the data that OET collects is freely available. So if there's a scientific purpose for it, it is it's available to scientists.
That's cool.
Yeah, that's a nice shot.
That's a
Look
good
at that
picture.
backdrop, eh?
Wow.
Answer the question from Cal again. Has anyone on the team ever eaten anything from the deep sea? I know Meredith has at one point on one of another expedition she was on. They had shrimp. They were crawling, so she was able to eat fresh shrimp from the sea. Look at that face.
That could not have tasted good.
No, you have to ask her. I think she actually quite liked it. So,
Oh my God, that
Following us for the light? Or
You're down.
it's full wide.
Cool.
Wow.
So, Phones in the chat says, this is the most active Chaunacops I've ever seen. Is it more active at shallower depths?
I don't know if it's really
Delta's at
all that
2-2.
much shallower than we typically
Down a
see
little
them. If we get any shallower,
Come back down.
maybe shallower than 1,000 meters, we start seeing its close relative Chaunax, which is slightly different morphology and color.
Bye, Chaunacops.
Hi, Chaunacops.
I personally like Chaunax better than Chaunacops. That's just my humble
Hot
opinion.
take.
That's just a Pokemon name there.
Chaunax.
We're just listing off.
What does it evolve into?
Chaunax evolves into Chaunacops.
Charizard.
I think it would probably go the other way.
Charizard.
I think it would probably go the other way. Yeah. Chaunacops is the diminutive.
Of Chaunax.
Yeah.
I think I like Chaunacops.
That
Chaunacops
one's
is definitely-
flatter.
You all don't know what you're
Coming around.
talking about. This is such
Coming
an
around
amazing
clockwise.
fish. Like, look at that.
That one's pretty good.
Oh no,
This gives the blobfish a run for its money.
that's true. It is a pretty ugly fish.
That's true. I also
mean,
do like
cute.
the blobfish.
Totally cute.
When we dive at Johnston, maybe we'll see chonax.
So, can the fish this deep see the brilliant colors that we're seeing here?
You know, I don't know if there's a lot of research that's been done on about deep sea fish or even deep sea animal eyesight. Most likely they're using other vision capacities than visible light spectrum, only because they don't have access to visible light necessarily down here. So they might have, you know, eyes that are either low light adapted, so they can see things like bioluminescence, or there's probably a very strong reliance on like tactile
Do you want to come up underneath?
senses, you know, sensing for vibrations or sensing for
Zoom in
movement,
there real quick, Dan.
Yep.
pressure waves, things like that.
So, why is there so
Giant-
much color?
Giant worm tube.
Oh. So why is there so much color with no natural light down here?
Yeah, I mean the color is still expressed
Okay.
by the animals themselves. It could be something in their diet. It could be something that is being expressed, you know, due to phylogenetic constraints or evolutionary constraints. It could be, you know, something that's expressed purposefully to act as a camouflage of sorts. You know, I think, especially with respect to vision, you know, we have a very limited capacity to look at things in other than the visible light spectrum, because we can only bring our lights with us, and it's possible to bring different kinds of cameras, like low light cameras, or maybe, you know, cameras that can see in other types of wavelengths, but we don't have that capacity on the vehicles right now.
We're talking about it though.
Yeah.
Full spectrum gamut.
So, someone from chat also asks, do you think that light on the ROV scares away animals when they are used to being in the dark?
Unclear. Unclear. Yeah, it's not. I'm not sure whether you know, maybe it's the sound or noise, or maybe it's the lights. Maybe it's the temperature of the, you know, the vehicle is slightly warmer than ambient.
Atlanta heading roughly 270.
Those look like some barnacles there on the rock surface.
Ooh, walteria.
For a few minutes.
Ew or ooh?
Ooh,
No.
I love walterias.
That's the sponge, right?
Yeah.
Eep.
There was a dive I was on a few years ago down in, I think it was in the very far south of Howland and Baker unit of Pacific Remote Islands, and there was just a wall, 10 meters, 20 meters tall, something like that. Just walteria sticking out vertically from the wall. It almost looked like you could climb it.
That's so
like,
cool.
Is that just like a big clump of their larva just happened to drop there?
Very favorable spot, I suppose. But walteria is very, very fragile. Yeah,
It's not rock climbingable.
not for seamount climbing or rock climbing.
Did you hear that, Dan? No bouldering on the walteria.
Right.
No. I keep looking at all these as just fun problems.
starting to see a little bit more.
More of a kind of a coral garden we got going on.
Yeah.
Yeah, we got a little bit higher densities here.
You want to turn the miter off and turn the bender back on.
The bender and ground fault sensor are on. You want to cycle them?
Oh, no, just turn the minute power off.
Hemicorallium, though.
Sorry,
it's all
leave
over
it
the
as
place.
is.
Very good.
No,
Very
turn
good
craft
site for
power off.
Hemicorallium.
Okay.
Yeah.
Just to get rid of that ground fault. I see.
Oh, excuse me. Okay, Lynette, we're ready for a move.
Lobster.
Lobster.
How are you seeing these things?
You should be able to bring your
Well, because I'm
hand out
not
there.
constantly typing you like you are.
Oh.
I'm constantly watching the screen.
All right, Atlanta heading 230.
So in this area we've got, yeah, what is this
It's
sponge?
like a brain.
What?
Brain sponge.
Is that what's actually called?
No,
No.
I don't. No, this is something different. This is not Polyapgon.
Spung.
Let's see if it's on our peripheran wish list. It's unusual.
Besides the fact that a sponge is spongy and coral has a skeleton, what are like major differences between the two? For like, is it how they eat, or how they reproduce?
Can you repeat that? I was working on something else back here.
Just asking what the major differences are between corals and sponges, besides their like hard skeleton in a coral versus soft in a sponge.
Yeah, I mean they have different feeding modes. I mean they're both feeding on things in the
meals.
plankton,
Right.
but corals are suspension feeders, so their food particles are free floating, and
Okay.
they are passively snapping up material as it floats by, whereas sponges will create currents to pump water through their
They actively, okay.
through their skeletal material and extract out nutrition that way.
Cool.
But
do
they're the simplest, the two simplest, or two of the simplest groups of animals.
Right. Do they reproduce in similar ways?
No, not really. I mean, they have both. Yeah, I mean, in the similarities are in that they both have sexual and asexual reproductive modes, but I would say it doesn't go much more beyond that. Sponges, it's very simplistic. Corals can sometimes have more complex reproductive modes after that. So corals can brood larvae, within their tissues, or they can broadcast spawn. Where I think sponges, their sexual reproductive mode is might be broadcast spawning, I believe, but it could be misinformed about that. We've had sponge specialists out here before, so we'll need to kick some of these geologists off and bring out a sponge.
Honestly, geologists, man.
Got too many of them. No, joking.
Answering the question in chat, how far away from you are you? Are you far from where the last dive was? There's a lot more. There's a lot of marine snow on that one. It does not seem to be much here. So I was wondering if there's a change of location or time that just change of location of time that meant less snow. We were about 50 nautical miles away from our last dive, according to the dive plan. So we are a little bit in a different location, which probably explains the not as abundant amount of sea snow that we see here.
I will say, this is anecdotally, but there's more particulate matter here than in our first few dive sites, though, the first 4 seamounts. Yeah.
And then another question. The Chaunacops and Chaunax are examples of what group? Like chimpanzees and apes are examples of primates.
Bony fishes, but their family is the lowest common taxonomic ranking, so the Chaunicidae family.
Chaunicidae.
Yeah.
According to Google, also known as the sea toad.
Oh,
Really?
coral
Yeah,
up there,
the
wow.
sea toad.
Sea toad.
I like that name. Sea
Yeah,
toad.
it's
So cute.
oddly fitting. Oh well, I just saw that your chat for disregarding that
Toads
question.
are cool.
But, yeah.
Atlanta heading 180.
So
Right.
regarding the anglerfish, would shrimp be its main source of food since there's not much else down here to ambush?
Well, not necessarily. I mean, shrimps, yeah, that makes sense. They're large, probably represent a good meal, but I don't think there's a whole lot known about what they eat specifically. We've never really seen them eat, but there's a great website for all your fish questions called FishBase, and you can look up almost every single species of fish that's known in the world and get an idea of, let me take a look at that purple thing and see if that's Victorgorgia there.
Victorgorgia,
And get any information
right
that's
there
been
for you.
available about its gut contents that have been conducted by scientists.
This is a cool overhang.
Yeah, that was
Just watching this ledge on my
Answer the question in
That's
chat.
over
Did
there.
I hear correctly that this 6th dive is the last dive of the expedition? No. I think we have a few more planned ahead of us. At least, I think at least 2 more, if not more than that.
as many as we can. I think we planned on something like 12 dives, ideally, but weather dependent. No, 13 dives. We planned 13 dives, I think.
Yes. And how many? We are on dive number
6, right?
On dive number 6. Toadfish, yes. Yes, it does.
Polyapgon, still mostly seeing paragorgias and Hemicorallium this area.
I
This
also
looks
saw
like, I don't know, probably looks like loose stuff, but it looks like it's all fallen.
oh, there's
this
an
pile.
anathemastes.
Yeah.
Well, we're coming up on a watch change, so
Okay, we can grab a rock.
whether we want to grab a rock now or let the next watch do it, we'll probably have to stop anyway.
We can grab a rock anytime, Steve.
We're,
Oh,
Ooh,
is that
Is that a
finally.
squid?
octopus?
no, it's a
Hey.
gumboat.
It's a what?
You
So
zoom in
it's a
there a
what?
bit,
That's a
please.
Dumbo octopus?
There you
That's
go.
an octopus. Wow.
Only ever seen the
It's
so
Dumbos in Nautilus
tiny.
live
Is that a
streams.
vampire?
No, it's a, well, they belong to the same group.
Oh, that's why I get confused.
Yeah, but this is a Dumbo octopus. First one of the cruise, I think.
I thought you said jumbo octopus.
Dumbo.
And I'm like, huh,
Yeah.
what a name.
Wow.
Dumbo
I didn't know they were so
octopus,
small.
got it.
It is really
Yeah.
moving it down slope there.
Wow.
Well, they can get quite large.
Oh yeah?
Yeah, the I don't know what this
Zoom out
is. If
just
this
a bit.
is
Yeah.
Yeah, no,
They're
get a
so
little closer so
cute.
it won't be so jumpy.
Right.
there's a number of different genera of Dumbo octopods.
Is another octopus behind it, or is that something else?
That looks like a sea anemone.
Yeah, I think that's an anathemastes along the edge.
Yeah, Anthomastus, yep.
Yeah.
Mushroom coral.
Anathemastes. There you go.
Nice flying, Dad.
Wow. Just floating
Yeah, I have to
there.
hold
Yep.
those in out
Yep.
because he's moving fast. I'll try and get
They
closer.
vibing.
Sure.
Oh,
I think his
Hi,
ear
Dad.
flaps.
it's so cute. I might have to change my favorite animal to this one.
Very cool animal.
I still think the Chanacops is going to be my favorite. So here's another question in chat. Do we ever turn off the lights from the ROVs to see
It's full
fish bioluminescence?
Try and get one more shot before he swims out of our tether range here.
Well, we don't usually
That's great.
see bioluminescence, but it's not because it's not happening. It's requires a very sensitive camera. It's a very
Delta's at
small
2-3.
amount of light.
Yeah.
All right.
Ah,
At the photons of light level,
Nice job.
and
Nice shot.
even a big camera
I zoom.
that we have wouldn't necessarily be able to detect it without some special tech.
so we don't possess that kind of camera technology?
We don't have low light camera technology on Hercules right now.
Oh well. So there's to answer your question in chat, Zed.
Oh, an eel.
Yeah.
Oh yeah, big Synaphobranchid. That's a huge one.
Synaphobranchid.
It's unusual to see them on such a steep slope. It's eerily unusual to see any fish on steep slopes like this.
Man, these heaves are a good 5 meters altitude.
What's that?
My altitude is oscillating around 5 meters or so, the boat.
Yeah, I'll try and
Well,
come and go back in front of you here.
Thank you all for tuning in to the 4 to 8 watch. This is Team Delta Dan and the Benthic Storms checking out for another watch change. Tune in another 12 or so hours and we will be back. I'm going to turn it over to Team You're Muted.
All right.
Still there, Dan? Oh,
Yeah.
oh never mind.
Good morning.
That's me out.
it's been pretty rocky, so I've been giving it an extra little bit of altitude and delta. Swings almost 5 meters sometimes.
Yeah, absolutely. And keep an eye on that sonar. Sonar looks nice and clear at the moment. But
Yeah.
I'll try and get back into a better spot here. Looks like there's lots of stuff to look at.
Oh yeah. Yeah, I'll swing around to starboard behind you.
Okay, sounds good. Look over here.
So we've been leaving the, we've been turning the bender and the ground fault sense off to run the craft.
Yeah. Yep. That's an old trick.
All right, I'm out.
Okay, thanks, Guzman. Will you zoom in, please?
That looks like part of that's been munched.
Morning.
All right, everyone.
Come wide, please.
The vessel is currently stopped, so we can explore, but just let me know when you want to keep going to waypoint 6.
Oh, we're stopped. Okay.
Yeah.
Well, I'm going to poke around here a little bit. There's a few things to look at.
Mm-hmm.
Let's look at this anemone. Yeah, you just missed it.
Yeah.
Will you zoom in, please? That's a big anemone.
Wow, yeah, it's like 20 centimeters. Nice.
Is that a little mushroom coral in the back there?
Yeah, it looks like it. And maybe a Paragorgia.
please.
Is that the same as a precious coral, the Paragorgia? That one?
No,
that's really
Paragorgia is bubblegum.
Okay, that's
Actually, that might
bubblegum.
be a precious though, which
Okay.
is Hemicorallium.
Yeah, I didn't see the chunks of
Nice.
bubble gum.
Yeah, those two look similar to me
Yeah, yeah, I have
sometimes.
trouble.
Yeah, I think we've been seeing a lot of Hemicorallium
Oh, okay, that's
on
probably
the
what it
previous
is
watch.
then.
Yeah.
Oh, cool. So, aloha kakayaka, awina la, ahi ahi. Good morning, afternoon, evening to our viewers out there. We've just switched our watch and you are with the You're Muted crew, who is officially unmuted, I think. So we're going to go through some brief introductions when we're ready, and then Rob is going to give us a little bit of a synopsis of what we're doing on this dive.
I'm going to take bubble real quick.
Yep.
All right. Take it away, Rob.
Okay, I'm Rob Piccolini. I'm the watch leader for this watch, the You're Muted watch.
Yes.
And I'm also the geology lead on this expedition, and I'm from the University of Rhode Island.
Thank you, Rob. Aloha, Liz.
Good morning. I'm Liz. I'm a biologist coming from University of Hawaii.
Aloha, Taylor Ann. Good morning.
Good morning. My name is Taylor Ann, and I am the science manager and data logger. And I'm also a graduate student at California State University, Northridge.
Awesome, awesome. Aloha, Emma.
Good morning, everyone. I'm Emma. I go to the Coast Guard Academy, and I'm the navigation intern.
Thank you, Emma. Good morning, Josh.
Good morning. Josh Chernov, Herc pilot for this watch and ROV operations manager for OET. I come from Vancouver Island, British Columbia.
Thank you, Josh. Aloha, Michael.
Good morning. I am Michael. I'm going to be your Atalanta pilot and assistant to Herc. I'm from California, Los Angeles specifically, and also a grad student.
Mahalo, Michael. Good morning, Dave.
Good morning, Dave Robertson, lead video engineer, and making all the cameras look good as much as I can.
You're awesome. Thank you, Dave. I'm glad we got to see a Dumbo octopus.
Rob, did you want to, the ship stopped at the moment, do you want to keep moving towards waypoint 2?
Yeah, let's keep going, please.
Sorry,
6.
6.
Yeah, let's keep going.
2 was last night.
All right.
That's true.
It's all melted together.
Good morning, Ivan.
Bridge nav.
Good morning, Ivan Vicente with the U.S. Fish and Wildlife Service.
Mahalo, everyone. Aloha mai. This is Hoku Pihana. I am from the Moko Keawe, the big island of Hawaii Nei. I am your science communicator for our You're Muted watch today. Thank you for joining us, and I'm excited to take a look at all the awesome things that we're finding down here at this unnamed seamount of number 13. And I think we're currently, yeah, it's been a really nice watch thus far. So whenever we're ready, we'll go through a little synopsis of what we're going to be doing today.
Yeah, I've been seeing a lot of these sea pens on this dive, on the rocks.
Are they sea pens?
I think they are. I thought I heard them say earlier, but
yeah,
the sun was kind of low.
They might be sponges.
Oh, yeah? Yeah, because I saw a lot of them on these little rocks that are isolated with a lot of corals on them.
Let's zoom in, please.
yeah.
I don't remember seeing these at any other locations.
No, me neither.
Yeah.
Yeah, I think those are
little sponges.
Yeah.
Yeah, that's cool.
Sea pens are more in the sand.
Yep.
But those are interesting. I haven't seen that type. Oh, a little cucumber.
Can you come down a bit? You're pulling on me every time I can't zoom in. Can we zoom wide, please?
Coming down.
Their sonar is like pretty much clear at the moment, so there's much danger to being a little lower at the moment anyway.
Yeah, we're still at unnamed seamount number 13. We're on the northeast flank, and we've been heading up a bit of a ridge for the last 12 hours or so, and about halfway through our planned waypoint dives. In the previous watch, did have a chance to spend some time in a pretty spectacular, coral garden on a little flat area just below us. And so we're going to continue up, and maybe if we get a chance to get to the top of the Gio, go across it and explore it a little bit.
That'd be exciting. This one has had a lot of really neat things to check out. So I'd like to open the floor. We do have our first question is actually geared towards Michael.
Well,
Michael, whenever you're ready, let me know.
Sure.
Okay. So how does one get into piloting submersibles?
Instagram. That's how I got in. I saw the
I love it.
the post saying that there was going to be the internship program for the 2020 year, and yeah, I just happened across it, and then I checked out the site and read up all about it, and then that's how I got in. And I think a while ago, in another watch, Josh had said something like, "There's no one way."
Right.
You know.
Yeah.
And so, like, having technical experience helps, I'm sure, and and things like that.
Sure.
But it's just a matter of looking into it and then just like and applying. And if like you have like no experience or you don't know how to get in, like just start contacting people. Just like, hey, I want to do this. How do I get to a point where I'm capable of doing it?
Yeah, good point. Thank you, Michael. I think that's, that's cool. It's just charging it and seeing what comes of it and asking the questions, being inquisitive.
So is that portion diseased there, you think, or?
I think it's been colonized by the zoanthids, the yellow part, and then the pink is the coral.
Wow.
And it's interesting. It looks like the brittle star prefers the injured part.
Huh.
Or maybe the brittle star caused the injury.
I was just going to ask that if that it could be one or the other.
So that's another Hemicorallium.
Looks like we have these Polyapgon sponges too. There's an old base that kind of.
Striking. Look at how tall they are. So our second question is how many shifts of different crews are there? So we have 3 shifts that rotate throughout a 24-hour period. Each of our watches are split in 4-hour shifts. So we rotate in our evening watches and our day watches. So you get a different flavor with each shift. We have the You're Muted watch, Sea Pickle watch, and what is the name of our Dan, the man in the it's the, oh, I'm going to make up my own name now. It sounds like a band. I hope we see a shark too. We did get a shout out. So hello, You're Muted watch. You're my favorite watch.
That's right.
Oh, thank you. I hearted that one.
Really?
Right. We were babies. Liz, I got a question for you. Coming up here, are the feather plants new coral growths? Or the feathery plants, excuse me?
They're corals. They're probably not very young, so I wouldn't call them new.
Okay.
There's another anemone there.
So in this case, size wouldn't necessarily suggest age.
Yeah,
given the environment.
Not necessarily.
Okay.
I mean, they will get bigger as they get older, but there are some that are going to stay small. Like this one. Well, one of the ones we're seeing.
Right. Yeah, I was just looking at that
they tend to be pretty small. I think that's maybe Bathypathes.
Nice. And our third watch crew is named Delta Dan and the Benthic Storms. See, I'll tell you, it's a band waiting to happen right there.
Wow. There's so much of that Hemicorallium.
Where's the Hemicorallium? The pink?
The pink, yeah, I think.
Beautiful.
Yeah, I think this is kind of what they've been seeing here and there on the last watch.
Wow.
Good overhangs with a lot of Hemicorallium.
We used to like partial zoom, zoom or whatever, kind of frame up that.
So we were able, so the Delta Dan and the Benthic Storms watch was actually the ones blessed with being able to capture the Dumbo octopus. And as we were all getting up and ready for our day, it was on screen, so we were able to see it a bit too. So it was very exciting. Hopefully we'll get to see more of them.
Yeah, that was less than an hour ago, and they'll be definitely including that in the highlights, so that it'll be posted soon on nautiluslive.org on the highlights. Yeah.
Looks like a big elephant ear, this sponge up above.
Oh.
Yeah.
Well, look at down there. The stop in the distance.
Oh, there's a lot of stuff down there.
Yeah, the previous watch noted that the current flow is low here, but it seems to be out of the south a little bit more than the north that we predicted from the models.
Interesting. Look at that too. Liz, is there a common name for this particular coral that we're seeing right now?
Precious coral.
Thank you.
That's awesome.
It's beautiful.
Yeah, nice shot. Nice captures, Liz.
Thanks.
Yeah.
It's all Josh driving. Just clicking buttons.
Oh no, it's a Kako thing. I've been going through all your images and you've captured some really nice ones. Yeah.
Okay, good.
So it was very helpful. You
Go,
guys are
come
doing an
wide,
awesome
please.
job with that.
There's a lot. There's a lot going on.
Yeah.
Yes. Yeah, there's a lot going on. So we were also blessed with seeing, I woke up to being able to see the Chaunacops this morning on the TV. And so I was really happy. It made my morning. So a lot of us were able to see the Chaunacops from the Delta Dan and the Benthic Storms watch. Okay.
This is almost like an overhang. Or am I just weird with my
No,
perspective?
It looks like overhangy.
So we just got another shout out. Shout out to my daughter, Coralie. We love watching all of the videos. Can't get enough. Keep watching. Thank you for sharing. And if you have any questions, always pop them in the chat. And pilots, I got a question that just came in for you. What is the new plate metal rigging on Herc's light bar? Oh.
Plate ringing on a light bar.
They might be seeing the Norbit multi-beam sonar, maybe. It's kind of sticking out in the front, in the middle. So that's a downward-facing multi-beam sonar. To working with Chris Krasnowski on developing some more high-resolution bathymetry data products that kind of help to increase some of the resolution in the areas with the other maps that we create from the ship, so we can kind of pinpoint some really, really, really high resolution areas by bringing the sonar actually right down to the seafloor.
Nice. Thank you. Thank you, Josh.
Okay.
Pretty cool spot.
Yeah, looks like there's a lot of old holdfasts too.
Wow, yeah. That's a beautiful view from Atalanta
So
right
we're kind
now.
of going across the hill right now,
Yeah,
so we'll just be careful about if we get a little close on the sonar, then I'll come up the hill and then across.
Okay.
Yeah.
I can also angle it a little more steeply if you want uphill.
No, I think this is cool, this area. So we just might have to come up a little bit if we swing a little too close to the wall.
I don't, yeah.
I think we're good right now. I mean,
I think we got at least 40 meters before that has to happen.
Yeah, we're all good at the moment, just in case we need to come up, we can just sort of rise up and cross-section a little bit higher up.
Yep. I've been wondering about the offset of the ship in Argus, or Atalanta and Herc. It seems like it's, the ship has been consistently to the south for most of this dive or this deployment. Is that an indication of, deeper currents maybe, or?
Well, the ship is moving kind of southwest, right?
Okay.
So we're going to trail behind in that way. And also, we're not going to lay back as much, only being in 1,800 meters.
Okay.
So when we're at like 3,000 or whatever, we're going to be way more laid back.
Because I'm always trying to find analog measurements of current.
The current's definitely, so when we're stationary and then where we sit, like when the boat's totally stationary and everything's all settled out, that will give you the indication of like the current, which direction that we're laid back. Okay. Then of course you have that cable through the entire water column, so it's hard to know. There's always going to be crossing currents and stuff on the different layers.
Yeah, well, we did some work along the equator.
Partial zoom, please.
With the deep counter currents and currents there. it was fun putting the CTD down and just seeing the whole thing kind of swing out when the rosette hit the currents. And then it came back down underneath the ship after when it was just the wire and the current. Oh, these are gorgeous.
Yeah.
So there's a question, how precious is precious coral?
I think, is it called that because that's the type that was originally collected for jewelry? Was that what Steve was saying?
I think that's right, yeah.
Ah, it's so precious.
It's all precious.
I wonder what they made jewelry out of it then.
Yeah.
So, okay, question. If precious coral is only found at so then I'm wondering, is precious coral found in shallower areas where it's more accessible to gather?
Yes.
Awesome. Okay.
Okay,
thank you. That's a beautiful sponge. I wonder what kind of jewelry they made out of it. Man.
Come up just a little bit.
So just a little bit of updates for everyone here, to all of our viewers out there. We're currently at about 1,823 meters. Our temperature is 2.34 degrees Celsius. We have an oxygen saturation of about 16% and an O2 concentration of 71 micromoles per liter, and our salinity is sitting pretty consistent at 34.6 PSUs.
What are these little things on here? Is that like a hydroid?
Can we zoom in, please?
Oh my goodness. Oh, it's on. Are we going to have the bug cooking show again?
Not anytime soon, no.
Oh, that was a limited run.
That was a special series.
Yeah.
Coming up with a new channel.
It's a Netflix special that is now departed. We did go into the dessert challenge.
Well, we are in negotiations for a spin-off.
Oh, we could have a spin-off, but what would we talk about? Would it be
Are they old
bugs?
You think?
Oh, that's pretty.
I don't know. They look.
Yeah.
What are those, Liz, that we're seeing right now?
I'm not sure.
Okay.
I think the small fuzzy things might be hydroids or bryozoans.
The little white ones. The yellow, it looks like a coral.
Okay.
Yeah.
No, it is hydrozoan.
Is it hydrozoan?
Yeah.
Okay. Thank you. I have a question coming in here. For our scientists, is this area part of a bigger reef system, or is it more of a standalone environment?
So we're on a seamount. Well, is this a geo or a seamount? Have we decided?
It looks like a geo to me, but it's been named seamount number 13.
Okay. Well, basically that means it's sort of a little bit of an isolated underwater mountain, but there are others in the area. So it's not directly connected to them, but there's probably still going to be exchange in terms of larvae.
Nice diversity here in this shot.
Yeah.
Can we zoom in on this yellow one, yellowish one?
Looks like another zoanthid situation.
Where?
Yeah, I'm just trying to see if it's a plex or a zoanthid one.
Oh, yeah.
I think I'm just really captivated by the sponges on this one.
Is that our favorite Victorgorgia too?
Yes.
Ooh, what is it?
Victorgorgia, the purple one.
Purple is Victorgorgia.
Yeah, Victorgorgia. Is it because they're so gorgeous?
And then, that looks like you've got zoanthids colonizing.
Well, that's interesting.
Really?
Yeah, so you can see the larger yellow polyps as the zoanthids, and then the smaller pink ones are the coral.
Okay, thank you, because I've been dying to ask you that for a while. I'm like trying to differentiate it myself, but then it always helps when you help us see it. Which brings on our next question, Liz. Is the marine snow more buoyant in the smaller particles you're used to?
They're asking if it's more buoyant?
Yeah, is the marine snow more buoyant than the smaller particles you're used to seeing?
So, generally speaking, smaller particles sink slower, so you could say that they're more buoyant. They're less heavy.
Okay.
So any particle that we can see with Herc is going to be relatively large.
Okay. Like what we were seeing on our last dive.
I think mostly they might be noticing that the current is less strong than it's been at previous sites, so they're not moving as quickly.
Okay. Thank you, Liz. I also have a question here for our pilots.
Yeah.
When the ROVs are on a dive, do you also control where the surface boat goes?
That's right, yeah. The boat is actually what actually puts Atalanta in the correct spot, which then allows Hercules to get to the right spot.
Okay.
So we have to start with the ship and then get Atalanta in the right position where we want to look, and then Herc kind of is a third piece of that puzzle that kind of follows.
Can we look at
Kind of
this
leads,
one?
but has to follow at the same time.
Okay.
Yeah.
Right, it's like a dance.
It is a dance, yeah. But it all starts with the ship.
Okay, thank you, Josh. Yeah, and for our viewers out there, if you switch to our Channel 3 feed, you can see the imagery that our navigators are looking at that help us really position and keep everybody connected with the ship, Atalantis, and Hercules there. And then, so aloha from the Netherlands. Mahalo for joining us. I'm going to pass this one to Liz again, I think.
Home of marble racing, evidently.
Oh yeah, marble racing. With so much precious coral there, can we expect some golems as well? Did I get it again? Did I get it?
It's a Lord of the Rings joke.
Ah, dang it. Why you guys do that?
That's good. Wow.
You're
Look at the
walking right into
diversity
that
in these fan corals.
I think that person could be a member of our watch, with bad jokes.
That was a good one.
unless
oh my God, I got it now. Okay.
Is that a squat lobster? That little guy?
Right down here, yeah.
Yeah,
Right there.
Maybe.
Can you zoom in, please?
Yeah, this is probably some of the best coral we've seen diversity in our watch.
Or it's pretty amazing.
Is it one of those hermit crabs that puts an anemone on it?
Looks like it.
Yeah, that's what it is. Hey.
Cool.
Weren't you saying you wanted to see one of those?
Oh, oh, that's so cool.
Oh my gosh, it has a little
so they attach an anemone to their shells.
Why?
That's their little thing. Isn't it like
I don't know. I think protection.
Protection.
I don't know much about them. That was great.
But isn't it like a mutualistic relationship too? Like both benefit from it or something?
Oh, there was a squat lobster too.
Oh, there was. Okay.
Oh, there was.
That was a white one though.
What is that? Oh, I thought that was a worm. Off to the right there, but it's not. Is it a tube worm? Okay, now I'm gonna have to really make sure I read through these questions.
No, I want to give a shout out to our scientist ashore, Asuka Matsumoto. She's doing a wonderful job helping us identify these various corals.
Yeah, thank you.
Yes, thank you so much, Asako. That's really helpful. I am learning a lot. Okay, so I think this might be a good question. Why are all these corals so vibrantly colored? Can pigmentation provide them with any advantage in the darkness?
Yeah, that's a great question. I
Yes,
don't
it
think
is.
there's really a clear answer to it. But yeah, so at this depth, you're not really getting sunlight. So, you know, these colors wouldn't be visible except for with bioluminescence. There's a possibility that there is some sort of fluorescence associated with them.
Okay.
It also could be that they're descended from shallow water corals that use these pigments and that they just haven't lost them.
Okay.
Cool.
Cool.
No Doppler right
Thank you, Liz. I'm going to pass this question over to Ivan. Ivan, what is an atoll?
Oh, I think all of us can actually define that, but there's one person in this group that can definitely give you the right amazing.
Okay, I'm going to toss it over to Rob. Rob, what is an atoll?
Well, it's the remnants of a sunken seamount.
Oh.
That has a coral reef at the top of it. And officially, the atoll is the fringing reef that's above water.
Mm-hmm.
But,
okay, before it's starting, like it's in its process of sinking.
Yeah, and sometimes it can have a peak in between of it with a surrounding coral.
Okay.
Or if the peak is now sunk all the way, it could just be the coral.
That's that fringing reef. That's
but they kind of call the entire seamount reef system an atoll as well.
And Rob, at what stage does an atoll stop being an atoll?
A little more.
maybe?
Good question.
It's the same thing, right? If it's
I
a
think
wall,
when it's
a wall,
totally
you gotta
underwater,
get in the
and
right
not
When it's
gro- not
completely
growing anymore.
No, I was saying
underwater,
the ship.
then it's no longer an atoll.
Yeah,
Come
well, I
down
mean, once
more away
the
from
the
the
coral
wall.
reef and
You
the
want
corals on it
follow
stop
along.
Oh, I
growing,
see, because it's kind of going
when
north
it's sunk
south.
below
And it could
the
go,
photic
like, a little
zone
more
of the
south.
area.
I think we'll
And
just,
and
we'll
then
just
when
come
does
up
it
and
become
then
an
we'll get a little
atoll?
further
Does
away,
it like
and then
at
we'll
a
just-
certain size or a certain
Is that
like
gonna
once
work?
the island is sunk to a certain depth, or
I think.
compared to like an island? Like I'm thinking of Midway
Because like if
Well, I mean,
the
if
wall's
it, I
right
mean,
here on Atalanta,
if it doesn't
right
have
here.
Yeah.
compared
a
to
fringing
Kauai.
reef,
We'll
Oh,
try.
okay, then it's
then
just
it's
an
just an
atoll.
island.
Whatever.
Okay. So those all playing together kind of
Yeah.
help define
Yeah.
its
I don't know
characters.
how the wall's going
And
to go
then I
in the
had
future,
another
so.
question. Do all seamounts, are all,
Yeah, we could
before
try
they're
that.
submerged, are
Let's
all
see how we do.
seamounts
It's-
seen
yeah, now
above
we're getting a
water
little further
first?
away.
Like
Atalanta's
with
getting
these
further away
guys, I
from
mean,
the
we're
wall
so
now.
deep. Would they have been like, I don't know what I'm trying to ask. Like, are they,
Yeah,
you
let's
know?
try this
No,
for a little
I mean,
bit.
some of these, I think the vast majority have never reached the surface of
Okay. Okay.
and so they're the ones who are, pretty much just a peak
Got it.
without a flat top.
Okay.
But then it doesn't mean that all the flat top seamounts have been
Yeah,
at sea
they're
level.
now 20 meters away.
Because
so
there
that's
are
better.
ways that you could actually have later eruptions that kind of make it a flat top.
That make it a
the-
flat top
look
wall
like a guyot.
underneath. Because see, that
I
was
don't
what
know
I
if
was
they
wondering, if a
almost
guyot
go
was
on top of the coral.
something that was already at the surface and then maybe had sunk. Or,
Yeah.
Please zoom in, please.
Thank you, Rob. Ooh, what is that? Is that a crinoid?
So that thing I
Yep.
called a hermit crab, it turns out it's actually a type of crab called a carrier crab. They're not true hermits. They have modified rear legs that carry that anemone around.
Wow.
It's so funny.
Very cool. Well, it almost
So-
looked like a spider for a second when I was looking at it. I was like,
Yeah.
wait.
These are gorgeous.
Yeah.
Oh, we got a shout out to Dr. Asako is the bomb.
So these I want to call Hemicorallium because they're more delicate.
Yep.
Okay.
Nice scenery you're giving us, front row.
Yeah.
Yeah, this is a
thanks.
beautiful.
Yeah, it's always fun swinging a 2,000-pound wrecking ball along the side of a big cliff wall.
Not stressful at all.
You have excellent driving skills. Oh, one question,
Oh, wow, that's
Liz,
cool.
that did come up in the chat was why aren't we seeing as much
There
marine
you
snow
go.
as we did
Right
in the
on the
last
sextant.
compared to the last
That's
time?
the one.
Nice.
Oh, sorry.
No, whenever.
Just give me a second.
Sure.
Oh, there's like a whole bunch on the bubble.
Yeah.
That's a sickly sponge back in the crevice there.
Yeah. So for the marine snow, I think what we were seeing that previous dive was a kind of a short-lived event. Isolated, probably small scale in both spatially and temporally. And so either it didn't happen here or it's already over. But that's my guess.
Yeah, I'm also wondering if the C4 morphology had an
Thank you,
impact
Liz.
Yeah.
too, where these are localized seamounts, where the previous one was a elongate ridge, like 100, 200 kilometers long.
Right.
step
Very
in the
different
water.
characteristics.
Oh, Atlanta's getting blown out.
Yeah. There's some sort of currents next to
We
that
did
cliff
see
wall.
some of that similar to try to slow her down, but not in the same quantities
Yeah.
we had seen
Yeah, that's right. Is that all coral right down there in the
Yep.
the depth?
Yep. Down here
Wow.
along the
before. Yeah. It's a sheer
lower
cliff.
left.
Wow.
Yeah,
It's really striking.
You can see the lava flows in this rock exposed. Yeah, these are look a lot like volcanics.
Oh.
Still.
Oh, goodness. So Liz, how delicate are the foundations of some of these corals that we're seeing? We're seeing a lot of the holdfast there.
That's a good question. I mean, they're strong enough to withstand the current. which at least in some of these seamounts can be pretty strong. But, I mean, you could probably pull them off their base if you wanted to. They're not super strong.
Yeah. Thank you. And then following that question, the person who had asked the age question initially had missed it. So how do you determine the age of a coral? Our coral expert has left the building.
Yeah, I don't actually know the answer to that. I know with some shallow corals, you'll get yearly growth rings, but that might not be the case when you get deeper.
Yeah, yeah. We were sharing that sometimes size and height may not be as indicative of growth in these regions, just given the environment.
You can have different growth rates depending on food availability.
Yeah. And then back to you again here, Liz. So were you able to examine the snow that you collected on the previous dive?
Yeah, I took a look at it. I wasn't able to reach any big conclusion about what it definitely was, but I'm calling it phytodetritus, so that's basically dead phytoplankton. It could have been a few different types of phytoplankton. Around here you get a lot of what you call cyanobacteria, so that's actually photosynthesizing bacteria. It could be large masses of that.
Okay.
Could be some forams like Rob had hypothesized. So
right.
Yeah.
Okay, cool.
I'll do some sequencing back on shore, maybe be able to pin down a little better what it is.
Oh, okay.
I mean, how do you tell the difference if it's a foram that has collected a bunch of phytoplankton in it as food?
Well, because some forams have mutualistic phytoplankton within them. But yeah, I don't know.
Yeah, so somebody made a comment just making the observation about the snow and how they're seeing some here too as well. And like Liz had mentioned, we did catch some of those larger marine snow that we saw at the last dive. So we didn't gather any for sampling, but we did notice that it was here too.
Yeah, so what we're seeing here is a little more typical of what we've seen on most of the other dives in this expedition. You're seeing something in the water, but it's not sort of large, abundant clumps.
Yeah, Josh, I know it's not ideal, but some point when we kind of get above here to grab a rock sample, we haven't got one in a while.
Okay.
Yeah, whenever you get caught up and everything.
I'm definitely caught up. Just not sure what's
available.
Might be.
Yeah,
if you see something that you think is sampleable.
Yeah, because morphology here is a little different than what we've seen before on some of these slopes, so I'd want to confirm the lithology.
Muted.
It's very steep.
It is very steep, yeah. Don't even have altitude. Doppler's not seeing anything.
Another Victorgorgia there.
Meters.
Zoanthid on a Paragorgia.
Yeah, it looks like it.
Yeah, it's going 0 to 27.
So we have a question coming in regards to the difference between coral colonies and the reference to coral gardens. And they're wanting to kind of know the difference. So the way that I understand it is, and maybe Liz, you can help me with this, because we've been using coral gardens in the sense of the abundance of corals on these rocks that are of different species.
So a coral colony is actually referring to what looks like an individual.
Right.
So like one fan that you're looking at, that's actually a colony of many organisms.
Come up a bit more.
Yeah, just come up a little bit more.
And then the garden itself is these,
keep it around 20-ish.
The abundance of the different colonies all together on the rock,
10.
or within a particular environment.
Yeah.
Thank you. And do we expect to see these seamounts eventually fully covered by different types of coral over time, or does that depend?
Probably not.
On the area, region, environmental conditions, because we were seeing, not the last dive, the dive before, the corals tended to gather on those big independent rocks that were. So is it?
Yeah, at this depth, you're not really going to see complete coverage of the rock by corals.
Oh, okay.
Especially because, when we think about corals in the shallow waters, a lot of those are those stony corals, which are the reef-building ones. These ones, most of what we're seeing are octocorals, which are soft.
For a rock sample?
Yeah, I think if there's anything in there, it looks like some of the stuff is loose.
I don't know.
Okay. Yeah, the only one that's kind of big there.
I think we might be able to do it on the move, but we may have to stop the ship because I can't obviously land.
Yeah, I don't think that's. Just keep going, find a better spot. Yeah, sometimes it's tough on these real steep scarps here to tell the lithology.
Huh? So somebody has officially requested our mosquito burritos recipe. If we have a mosquito burrito recipe, feel free to share.
All right, what you do is...
Oh, no.
This is how you do it.
If we have one, I don't want it.
Thought we decided that that bugs were off the table, as it were.
Right, that's a different show.
We've put that in the repast.
Yes. We've canceled that series. Okay, so this question I'm going to gear it towards Rob. When you're ready, let me know.
Yeah, go ahead.
Do all types of lava rocks eventually will all types of lava rocks eventually be covered by a manganese crust? Oh boy, we got a lot more orders coming in.
Yeah.
Oh my. If it's submerged long enough in the ocean, how about pumice?
Yeah, it sure seems like that's the what we're seeing.
We gotta zoom in and kind of frame up that little patch there.
Yeah, it's awesome.
Oh, okay.
So would that be a plexaurid, maybe? This yellow one?
Oh, yeah.
Oh, that's pretty.
We'll zoom in some more, please.
So Taylor Ann, I have a question for you. This was asked yesterday, but the person who had asked it didn't get to hear the answer.
Doesn't like a plex.
And that was, how many samples can you collect?
And little Chrysogorgias around it.
In any one dive? And how do you decide what to collect?
So antidot.
Yeah, so we can collect quite a few samples. On our starboard side, we have a bio box with 6 partitions. And in the front, we have a larger box with 2. And then we have 7 slurp jars where we could put samples in. But we can also layer these samples depending on what species they are, what type of sample it is, and what we're trying to do with them. And then we also have 6 niskins and currently 3 push cores, but we can fit up to 5, I believe.
Wow.
And you can even put more on if you switch things around on the ROV a little bit. But yeah, we can collect quite a bit of samples. Just depending on how long the dive is, we have to plan accordingly of what we're collecting.
Cool. Thank you. Thank you, Taylor Ann. Oh, boy. So we just got to notice that they had blueberry bagels with cream cheese smothered with ghost pepper sauce and a beer brat with sweet hot mustard. I think we've opened... That led me into the donuts. Does anybody have a favorite donut they like to eat?
There's a lot going on in that meal, though. I mean, blueberry.
Right? That was action.
Bagel.
Somebody wants some burgers and fries.
Sauce, brat.
Brat. Wow. Brat. What's a beer brat?
Bratwurst.
Bratwurst. You make those.
I make sauce. I make kielbasa.
I was close.
I put all these different corals are different colors. So cool.
I would like a burger and fries. That would be good. So my favorite, has anybody ever gone to the Voodoo Donut Shop in Oregon?
Please zoom in, please.
Yes. It's a mushroom coral too.
Ooh, mushroom coral. What is that?
Mushroom, it's off now, but...
Just off screen to the right now.
Okay, I guess that's another Hemicorallium.
Yep.
Looks like it's part of its little dead.
Yeah.
And then
that one sticking out right there?
There's the mushroom coral there. Now you can see.
Oh, wow.
Yeah. Looks like a big one. And there's just like little Chrysogorgia like all over the rock.
Oh, yeah.
Do we do a Niskin?
Come wide, please.
Do you know when they last did one?
I think they have 2 left.
2 left, yeah.
Yeah, they have 2 left. Yeah,
we
so
can
might
save
wanna
that maybe.
Wait.
Yeah. Once we get up toward the top here.
Yeah. Oh, they did one just before the watch change. Okay, cool.
Oh, good. What is that right there?
What is that?
Is that another Bracingid?
Yeah, maybe.
Or it kind of looks like a crab also from here.
For a second I thought it was a sea spider, but I think it is a Bracingid.
And we have a little cave here.
Oh, yeah.
Off to the right.
Boston cream donut. Oh, that's so good.
Oh, zoom in, please.
I really like the maple and bacon donut. So good.
Yeah,
I haven't
it's another
tried
one
the
of those
Fruit Loops.
Bracingid stars.
There's a Fruit Loop donut.
They're very weird, the shape of their arms. They've got like those 4 long arms.
Hey,
is it possible to come right a little bit, Josh? There's a little cave there, a little inset.
So Liz, did you guys determine what that was? Was it a sea star?
Yeah.
It is. And so it just has...
Ooh, cool.
Yeah, no, if you zoom out
to
the right, there's a little bit more.
Yeah, there are some caves.
I thought I saw critters in there.
Ooh.
Yeah, this might be limestone now. This might be the reef material, yeah, up there, just above.
Wow.
Just this little spot right here?
Yeah, up above, yeah.
Oh, cool.
I'll zoom in.
Wow.
Yeah.
Is anyone in there?
I don't see them.
What is that?
Yeah, this could be limestone now. This could be part of the reef material.
Oh.
Usually don't get that sort of feature in lava.
Is that difference in tones of color, like that gray and ashy looking color?
Well, I mean, it's just the morphology. I'm not seeing any real pillows or lavas. That the limestone is much more angular.
I see.
And it may be that this whole steep scarp has been this limestone reef material.
Can we look down here?
Maybe tough.
Yeah, it's okay. Wait.
Yeah, there's a lot more of these embayments in the scarp.
Zoom in, please.
That yellow one's a little different too.
Yeah. And
that doesn't really have the plexaurid shape.
Oh, that's neon yellow. It's cool.
Yeah, I'm not sure what that one is.
We haven't seen one like that before, have we?
I don't think so.
Oh.
Yep, come wide.
Asco calls it a metallogorgia.
I think that was the lower one.
Oh, okay.
The like finer one.
Okay.
So, Liz, we got another question coming in here. Are mushroom corals true corals?
I think they're also a type of octocoral, which I believe octocorals are true corals. They're still corals. They're not hard corals. They're not stony. They don't create a skeleton.
Yeah.
But they're still corals. They're just a different type.
Thank you. And when was the last time we found a lobster? And are they indeed immortal?
We saw a squat lobster recently.
Am I getting suckered in again?
No?
I don't know. That's...
We had a little teleportation session going on a minute ago.
Thank you, Liz.
Hi.
Yeah, Asco said that the that yellow coral was not plexaurid. It's something different.
You can plug in right there.
Oh, I do. I have a favorite coral. Does anybody have a favorite coral? That's the next question that came in.
I think at the moment it's the Victorgorgia, just because it's the most different.
So you're talking to...
I like the bubble gums.
So you're listening to video, this is the top row, you're listening to video.
Rob, do you have a favorite?
Yeah, I'm a Victorgorgia fan.
So video, co-pilot, pilot.
Yvonne?
And then you're listening to the bridge.
Generally speaking, I love the elkhorn, corals, and the current.
Oh, you do?
Yeah.
That's nice. Mine is Pohakupuna. Okay, Paragorgias lobata.
And if you can
I just love them. They're so beautiful.
Those are the ones that are really common in our reefs, right?
Yeah. They have a specialist relationship with a particular nudibranch that contributes to their reproduction.
Now you're muted totally. And then that's
plus in Hawaii we recognize our Pohakupuna as a real foundational organism of establishing identity.
You're not too far off.
So we're literally swinging along a wall.
It's pretty cool.
And then we're trying to keep Atalanta to about 20 meters off the wall. That's this sonar on that side. That's Herc. So Atalanta, Herc.
That's the real game.
And then if we get Atalanta to within 10 or so, then we could just come up, right? And then get a little further away. And then I just kind of choose a different path.
All right. We got, for anyone, what is your favorite fish? Uhu. I love uhu. That's a parrotfish.
No, not to eat, right? Just
I'll eat them too. They're good. I'll eat them. No, I really have an affinity for the uhu, the parrotfish.
They're great.
Yeah. They just seem to be the ones that come around and hang out with me when I'm diving.
Is that a spiral coral? I can't tell.
It looks like it. Which, was that the Irradogorgia?
I think so.
Yeah, so that's what I was saying.
We can zoom in, please.
Is this the one that you call the firework coral or the fire? Is it the fireworks coral? Is that am I?
We were calling it that, yeah.
Okay.
Our backup navigation system.
I was just looking at the highlights. There was a fantastic picture of it.
Oh, come wide, please.
But it was like coming in from the front, so you couldn't see it from this side view.
Is that any fidget gadget over there?
No, it's our backup nav system.
Oh, backup nav.
Yeah.
Oh. I gotta get a little.
Take a look at that one. There.
You have to get a mini compass.
I think it's their plex.
Yeah.
Yeah, I don't know if it's zoanthid or a plex.
Usually with the zoanthid, I feel like you can still see a little bit of the underlying.
Yeah.
Oh, zoom in, please.
Type.
Little fish there too.
There was a fish?
Yeah, it just went off the screen up our left.
Yeah, it looks like the plex.
Yeah.
Definitely some swirly current along here.
Yeah.
I think
Come wide, please.
It's kind of cool how it seems to be going up the cliff a little.
Going into the wall.
Okay, we'll come up a little bit.
It almost looks
not
like
too bad.
these are
We're
pillows
just under
here,
20,
or are they just
so we'll come
big
up a little
chunks of
bit here.
limestone?
Well, just the trajectory I was going in.
Yeah. Let's come up a little higher.
But the steepness looks definitely much more like a limestone reef fish. Ooh, that's gorgeous.
Wow.
One.
Feel like a partial zoom there, Dave?
Picture? Still, maybe?
Hmm?
The still?
Oh.
Just wonder if it's...
Oh, yeah.
I like the coral with the zoanthids because it like goes from two different colors. It's kind of cool.
Yeah. Another little cave almost underneath there.
Yeah.
It seems as though the zoanthids might prefer the Paragorgia.
Seems like that's mostly what we see them on.
Yeah.
Yeah,
I wonder why that is.
Yeah.
Must have some sort of contract.
I don't think that the Paragorgia's getting a very good deal out of it.
I don't think they're too happy with that.
Are these long ones bamboo corals, Dean? Have we?
This one here?
Yeah.
Zoom in, please.
I think so. Single unbranched.
I'm not seeing sections, but
Yeah, sometimes they can be a lighter color, but yeah, I'm not seeing them either.
Oh, maybe.
Oh yeah, I think I see one. Sometimes Steve said they can be like a bronze color instead of that dark brown.
Yeah.
But
thanks.
Oh, come wide, please.
Well, we're supposed to be going to waypoint 6 and 7 down here, but the wall kind of is running this way, so we're going along the wall. And you can see like the way the wall's running, and then imagine it's going like that.
That was another one of those yellow that don't seem to be plex.
See? All the way down there.
There's a nice wall of them
That's
here.
I'm always trying to stay a little bit ahead of Atalanta, just like in this kind of position here. And then right now, like if we're tracking DVL, I'm not getting my DVL because it's so steep. So this could be a little off. This is the...
I turned it to USBL. Otherwise it just would be back there.
Yeah, so we're actually following. We can change the source of where Herc is being driven navigationally from either the USBL system on the ship or the DVL. So when we're in a situation where this is my Doppler,
oh yeah, there's
I need
another
4
one.
beams to get a lock.
Question.
Josh,
And
chance
then,
to take a look at that one
if you're,
there.
because it's so steep, I'm not getting bottom lock, so we can just switch.
Oh, that's an anemone on a crinoid.
Right. That's cool.
Oh, look at the...
It's really bent.
Yeah, it is. I thought at first it was two crinoids sitting next to each other. Is that fly trap anemone again? Those are really neat looking. Oh, that's kind of a nice view of it because it shows you from the bottom, the underside. Oh, that's cool. So Liz, I got a question for you.
Anemone goes on top of the crinoid.
So all the corals seem to have a white base. What is that base composed of?
So most of these are octocorals, so they don't have a hard calcite skeleton.
Okay.
Or calcium carbonate. So instead, for octocorals, is it made out of like a protein? Do you know, Taylorian?
Yeah, I'm for sure that black corals do. I'm not sure what protein exactly that these types of corals have. But I know that's one thing that's unique for black corals is that they sequester a certain protein that makes it black.
Quite a bit.
Oh, okay.
So it's that skeleton it's flexible, but you know obviously strong enough to give them the structure to stand up. But yeah, not stony, not hard.
Not hard.
So.
Thank you. And then another question here in regards to bioluminescence is, are you able to see it even with the lights on?
No.
No, so it's really just...
It's pretty dim relative, very, very dim relative to the ROV, and so to look at bioluminescence, there are special cameras for that. It's another mushroom coral.
Thank you. And then